We often talk about the human body as if it were a car engine. The assumption is that decades of movement, blood flow, and physical existence simply grind down our gears through mechanical friction and use. But biological parts don't actually wear out like metal ones. Your body exists in a state of constant, active reconstruction. Your entire skeleton, for example, replaces itself completely roughly every 10 years. Curious. This creates a paradox. If your physical components are getting swapped out for brand new materials on a regular basis, why do we still end up with brittle bones, fragile skin, and failing organs? This suggests that physical deterioration might not be a failure caused by the environment, but a limitation built into the internal mechanism of repair. To understand why our bodies eventually shut down, we have to look past the macro level of our limbs and organs and analyze the precise cellular engineering of how we heal. The foundation of this repair process is the stem cell, a biological blank slate. Most of your cells are specialists. Once a cell becomes heart tissue or bone, it is permanently locked into that single function for the rest of its life. Stem cells possess pluripotency, the unique capacity to transform into almost any type of tissue in the human body. When damage occurs, these blank slates read chemical signals. They migrate to the injury, physically transform their structure, and divide to rebuild the missing tissue. With this level of microscopic engineering, the body possesses a self-correcting system far more robust than any human-made machine, raising the question of why this process ever stops. The flaw is found in the replication process itself. Every single time a stem cell divides to heal an injury, it must make a perfect copy of its DNA. At the very ends of our linear chromosomes, we have protective structural caps called telomeres. These act like the plastic aglets on the ends of shoelaces. They prevent vital genetic code from fraying or degrading during cellular replication. But there is a hard limit to this protection. Known as the Hayflick Limit, research shows a human cell can typically only divide about 50 times before the telomere fuse runs out. Once the telomere is gone, the cell is exposed. To prevent catastrophic mutations to its DNA, the cell must permanently lock down and stop all future division. This locked, non-dividing, zombie-like state is what biologists call senescence. The steady accumulation of these senescent cells throughout our organs is the mechanical reality of what we experience as aging. This leads to the ultimate engineering question. Is it biologically possible to rebuild that burned-down fuse? Biology utilizes a specialized enzyme for this task, called telomerase. It has one specific job: to physically restore and lengthen shortened telomeres. We know this override is possible because nature already uses it. Organisms like certain lobsters and jellyfish constantly produce this enzyme in their cells. Because these animals can continuously rebuild their genetic fuses, they are functionally immortal, proving that the biological clock can be bypassed. If the mechanism for indefinite life exists, why hasn't human biology evolved to use it? There is a terrifying biological trade-off. If a human cell mutates and accidentally keeps its telomeres active, it will ignore every stop signal and divide endlessly without dying. This state of immortal cellular division is the clinical definition of cancer. This places regenerative medicine on a knife edge. We have the theoretical capability to master stem cells and regrow limbs, but the margin for error is non-existent. Ultimately, achieving biological immortality will require us to master the same biological mechanism that currently guarantees our death.